In this video, we have a look at how to select an appropriate multivariate analysis or machine learning method. Note that I will here cover only the multivariate methods that we have discussed so far. To learn more about the individual methods, have a look at my homepage where you can find videos that explain each method. We'll first get familiar with the iris dataset and then see how to plot this data and discuss how to identify outliers in multivariate datasets. Then we will see how to compute correlations and how to analyze multivariate data with PCA and clustering. We will also discuss how to select an appropriate classification method and finally discuss some multivariate statistical methods and regression techniques that are useful when dealing with multivariate data. For most examples shown in this video, I will use the classical iris flower dataset, which consists of samples from 3 different iris species. The data consists of measurements of the sepal length, sepal width, petal length, and petal width. These 4 variables were measured on 50 setosas, 50 versicolors, and 50 virginicas. The iris dataset looks like this. In total, we have 150 rows, where each row represents the measurements of 1 iris flower. These columns show the sepal length and the sepal width, as well as the petal length and the petal width in centimeters. The last column shows the 3 species, setosa, versicolor, and virginica. The methods that we will discuss in this video work for most types of multivariate datasets that include at least a few numerical variables. For example, instead of flowers, we could imagine that we have 150 individuals that have been divided into 3 different groups, which for example could represent 3 different treatment groups or 3 different types of diseases. The variables might represent measurements of, for example, blood pressure, cholesterol level, and concentrations of different proteins in the blood. For most multivariate methods, such as clustering and PCA, it is common that we first standardize the variables. To standardize a variable, we subtract the mean of the variable from all the values and divide by the standard deviation. For example, to standardize this value, we plug in the value here and the corresponding mean and standard deviation of the sepal length and do the math. We see that the standardized value is about negative 0.9. A standardized value of negative 0.9 means that the first iris flower has a sepal length that is 0.9 standard deviations shorter than the average sepal length of all the 150 flowers. In comparison, flower number 102 has a petal width that is 0.92 standard deviations longer than the average petal width. One of the first steps in analyzing a multivariate dataset is to plot its numerical variables to get a feeling for the distribution and to detect possible outliers. This can be done with, for example, a parallel coordinate plot. Every line in this plot represents the profile of each of the 150 flowers. The colors indicate which species the flower belongs to. This plot shows that the profiles of the 50 setosa flowers deviate a lot from the profiles of the versicolors and virginicas. This is mainly due to that the setosa flowers have a relatively short petal length compared to the other 2 species. Another simple plot that we can use to analyze the distribution of the numerical variables is a box plot. By using this type of plot, we can identify 4 outliers. However, the problem of using box plots to detect outliers is that we only study 1 variable at a time. This is how a scatter plot would look like if we plot the petal length versus the petal width of the 150 flowers. Each point in this plot tells us the petal width and the petal length of a certain flower. For example, the petal length of this flower is about 3 centimeters, whereas its petal width is about 1 centimeter. Now, suppose that there would be a data point down here This data point should be considered as an outlier, because it is quite far away from the other data points. The problem is that a box plot will not identify this flower as an outlier, because its petal length does not deviate much from the other flowers, which is true also for its petal width. What is strange about this flower is that it has a much longer petal length relative to its petal width. To identify an outlier like this, we can use the Mahalanobis distance, where an error ellipse is calculated. Data points outside the ellipse can be considered as outliers. Mahalanobis distance can also be used to identify outliers in more than 2 dimensions. We'll now discuss the importance of using colors when we plot the data. This plot shows that there is a cluster of flowers down here. To understand why there is a cluster down here, we can try to add colors to the data points according to some categorical variable in our dataset. In our example, we can add colors to the data points based on the species, like this. By adding colors to the plot, we can now easily see that this cluster consists of setosa flowers, which have a shorter petal size compared to the other 2 species. We can also see that the flowers of type Virginica have the largest petal sizes, whereas the versicolors are in the middle. In addition to plotting the data, we can also compute the correlations between the numerical variables. Since we have 4 numerical variables, the corresponding correlation matrix will be a 4x4 matrix. We see that the strongest positive correlation can be found between the petal length and the petal width, whereas the strongest negative correlation can be found between the petal length and the sepal width. We'll now see how to calculate a correlation between 2 sets of variables. For example, the combination of the 2 variables sepal length and sepal width can be considered as sepal size, whereas the combination of the other 2 variables can be seen as the petal size. To compute the correlation between the 2 sets of variables, we can use canonical correlation analysis. The correlation coefficient between the 2 sets of variables is here 0.94, which indicates that there is a strong correlation between the 2 sets of variables. We can also compute a scatter plot matrix, where we make a scatter plot between all numerical variables. For example, this plot shows the petal length versus the petal width of the 150 flowers. We can see that there is a positive relationship between these 2 variables, because a flower with a large petal width is likely to also have a long petal length, whereas a flower with a short petal length is likely to also have a small petal width. As we have previously seen in the correlation matrix, there is a strong positive correlation between these 2 variables because the correlation coefficient is close to 1. Note that the plots in the lower triangle show the same thing as the plots in the upper triangle. To avoid confusion, we should therefore focus on either the upper or the lower triangle. The same is also true for the correlation matrix, which is the reason why the cells in the upper triangle are usually left empty. Note that the correlation matrix and the matrix scatter plot are related. For example, the weakest correlation in this matrix can be found between the sepal length and the sepal width, because there is no clear trend between these 2 variables. The sepal length can, for example, therefore not say anything about the sepal width. From the correlation matrix, we can see that there is a negative correlation between the sepal width and the petal length. If we add colors to the points based on the species, we can see that the setosa flowers are down here. This tells us that the negative correlation is probably due to differences within the species. If we instead would analyze the correlation separately for the 3 different species, we would see that there is now instead a positive correlation between these 2 variables for all 3 species. This shows the importance of also analyzing the scatter plot before we draw any conclusion from the correlation coefficients. Plots and tables like these are only useful when we have quite few variables. Suppose that we would have more than 100 variables to analyze. A matrix like this would then be almost impossible to read. It would therefore be nice if we could combine several scatter plots into just one. This is exactly what we can do with Principal Component Analysis, PCA. PCA is used to combine variables so that we have fewer dimensions to analyze. If we would use PCA to combine these four numerical variables into just two, we can plot these two principal components against each other. Each dot in this plot can then be seen as a combined measure of the size of each flower in relation to the other flowers. If we put labels on the points that show which flower it is, we can see that, for example, flower number 42 is quite far away from the other flowers. This is due to that this flower has a quite unique profile. In comparison to the other setosa flowers, it has a relatively short sepal width. If we would have measurements on, for example, cholesterol level, BMI and blood pressure of instead 150 patients, we could say the patient number 42 has a health profile that deviates from the other patients. By using the variable species, we can add colors to the plot. Like this, where we now can see that the species are well separated when we combine the four variables into just two. In addition to the PCA plot, we can group flowers that have relatively similar sizes with hierarchical clustering. I've here used the Euclidean distance based on the standardized data with complete linkage. If we, for example, zoom in on the dendrogram here, we can see that the two flowers 118 and 132 have very similar values based on the standardized data, which means that they have a similar profile. This is exactly what we see in the PCA plot. We can also see that there is one setosa flower that does not cluster with the other setosas, which is consistent with the PCA plot where this flower is far away from the other setosa flowers. From the dendrogram, we can see that this setosa flower forms a subcluster with four versicolors. It is common to make use of both clustering and PCA to get a better understanding of the multivariate data. This is the correlation matrix that we discussed earlier. In association with this correlation matrix, we can compute hierarchical clustering based on the numerical variables of the standardized dataset. We can see that the petal length and the petal width form a cluster, which seems reasonable because these two variables show the strongest correlation. The variable sepal length cluster together with the petal length and the petal width, which also makes sense because the variable sepal length has relatively strong correlations to these two variables. Dendrograms like these can therefore help us to identify clusters of variables that correlate. Note that we can even use a distance measure that is based on the correlation coefficients to generate a dendrogram, which we discussed in the video about hierarchical clustering. So, is there a way to combine the dendrogram that shows the similarity between the flowers with a dendrogram that shows the similarity between the numerical variables? This is exactly what we can do with the Heatmap, where we cluster the rows and the columns of the data matrix. These two dendrograms are identical to the ones we saw earlier. A blue color represents relatively low numbers, whereas a red color represents relatively large numbers. The reason why the bar spans between -2 and +3 is because we here plot the standardized values, which span in this range. For example, we can see that the setosa flowers have relatively large sepal width compared to the other two species. What's nice about this heat map is that it can explain why, for example, two flowers form a cluster. For example, from our previous PCA plot, we know that the two flowers 118 and 132 were close to each other. These two flowers therefore form a cluster in the heat map. This is due to that these two flowers have a relatively large sepal width compared to the other flowers of the same species. By using the heat map, we can now therefore explain why these two flowers are quite far away from the other flowers in the PCA plot. Because the profile of these two virginicas deviates from the other virginicas because these two flowers have a relatively long sepal width. This can be seen also in the previous profile plot, where the two virginicas have a sepal width that is more related to the sepal width of the setosa flowers. We can also see why flower number 42 is far away from the other setosa flowers in the PCA plot, because its sepal width is much shorter relative to other setosa flowers. Another common clustering method to use is the k-means clustering. This method is especially useful when we like to divide the data into a predefined number of clusters. Suppose that we like to identify two clusters based on the 4 standardized variables. Then we set k to 2 and run k-means clustering, which will divide the data points into the following two clusters. In this case, we will put all the setosas into one cluster and all the flowers of the other two species into the second cluster. If we instead set k to 3, we will identify the following 3 clusters, which almost resemble the 3 species. If we instead set k to 4, we will identify the following 4 clusters, where the setosa flowers are now divided into 2 separate clusters. We'll now discuss the difference between unsupervised and supervised machine learning. When we use clustering and PCA, we do not use the information about the species to separate the 3 groups. These methods are therefore called unsupervised methods, because we do not tell the methods to cluster the data based on this information. We have so far only used the variable species to add colors to the plots. Remember that when we use PCA, we combine the variables in a way so that we maximize the variance of the combined variables. We see that the 2 species, Virginica and versicolor, are not well separated. If we instead would combine the variables in a way so that we instead maximize the separation between the 3 groups by using linear discriminant analysis, LDA, that will result in a better separation between the groups. LDA is therefore a supervised method, because it uses the variable species when it is trained, so that it combines the 4 variables in a way so that it maximizes the separation between the groups. This explains why LDA results in a better separation between the groups compared to PCA. When we use supervised machine learning methods, we will also generate models that can be used for prediction. For example, the first linear discriminant function of the iris dataset can be defined like this. Based on the scores of the first discriminant function, we can decide cutoff values that can be used to separate the 3 groups. Suppose that you would find an iris flower and that you like to know if it is a setosa, versicolor, or virginica. To predict its species, you measure the sepal length and the sepal width, as well as the petal length and the petal width in centimeters. You then plug in the measurements in the equation and do the math. We see that the discriminant score is negative 3.1, which is located between the 2 cutoff lines. The model therefore predicts that this iris flower is a versicolor. These kinds of models can be used to, for example, predict diseases based on a set of clinical variables. In previous videos, we have covered a range of different supervised machine learning methods. So, which method should we select? Suppose that we would have a dataset like this that might represent 2 variables that have been measured on patients with a certain disease and a number of healthy individuals. Based on this data, we like to create a model that can predict if a person has the disease or not. The models were first trained on the data, and the accuracy was then based on the test dataset with a similar structure as the training data. Classical methods such as logistic regression and LDA, as well as linear support vector machines, will fail to make a good job in discriminating between the two groups, because such methods rely on that the groups can be separated by a straight line in a two-dimensional space. A decision tree will perform better because it can create a tree that isolates the data points in the middle. Since random forest is based on decision trees, it will separate the two groups in a similar way, but usually with a higher accuracy. Naive Bayes will also perform well. The KNN method would be perfect for this kind of data, because it will classify based on the majority class of the K closest data points. For example, this new observation will be classified as having the disease, because the majority class of, for example, the 7 closest neighbors are of class disease. A support vector machine with a radial kernel performs much better than a linear support vector machine when the data cannot be separated by a line. This is also true for artificial neural networks in comparison with logistic regression. As we discussed in a video about neural networks, the hidden layers can create a much more flexible model compared to logistic regression. In comparison, when the two groups can be separated by a line, the classical methods such as logistic regression and LDA now perform very well. A linear support vector machine now has a similar performance as a support vector machine with a radial kernel. Although a decision tree performs quite well on this type of data, it will create a quite complicated tree because it relies on only vertical or horizontal lines to separate the data. Naive Bayes is clearly not appropriate for this type of data because the variables are highly correlated. We can therefore conclude that the performance of the methods is highly dependent on the structure and distribution of the data. So, which method should we use? If we have plenty of data, we could split the data into 3 parts by some random method. We can then use the training data to train the different classifiers, which means that the parameters of the models are optimized so that the models classify the data in the best way. Then we use the validation dataset to select the best model to classify the data. This means that we will use our models with estimated parameters based on the training data to see how well they predict based on the validation dataset. We could, for example, calculate the AUC value or the accuracy to compare the models. Finally, to determine the true performance of the best model, we can use the test data to calculate, for example, the accuracy or the AUC value of the model that was selected. Although more advanced methods like random forest and artificial neural networks usually perform better than simpler methods, it is very hard or sometimes even impossible to understand how they predict. If the user needs to understand how, for example, the model predicts a disease, a decision tree is probably the best alternative because it clearly shows how the decision is made. In comparison, it would be almost impossible to understand how a random forest classifier with thousands of trees would predict the disease. Models based on logistic regression and LDA can provide fairly simple equations that can be used for predictions. Such simple equations facilitate the sharing of the models in the scientific community. Another nice thing with logistic regression and LDA is that they are based on a simple cutoff value for binary classification, which can be manually changed by the user to increase sensitivity or specificity, or to compensate for an unequal sample size. Another advantage with decision trees is that they can handle variables on different scales without the need to first transform the data. For example, a decision tree can include variables on a numerical scale, such as age and the concentration of the prostate-specific antigen in the blood, as well as variables on a categorical scale, such as the output score from an MRI scan. Similarly, since a random forest classifier is based on decision trees, it also works well with different types of data. However, a random forest classifier generally performs better than single decision trees. Also, a random forest can be used for feature or variable selection. If the accuracy of the classifier is most important, I would select a random forest classifier. But if the understanding of how the classifier predicts a disease is most important, I would select a decision tree. We have previously seen that some machine learning methods perform much better than other methods on certain types of data. However, in most real-world clinical example data, there is usually a substantial overlap within the groups due to large individual variations. Although advanced methods, such as artificial neural networks, can separate the two groups like this, in comparison to simpler methods such as logistic regression and LDA, advanced models usually tend to overfit, which means that if we remove the training data and add a test dataset, the advanced model does not perform better than a simple model. Although the curve could separate the training data completely, it fails to do so on the test data. If you like to learn more about how different machine learning methods perform on clinical data to predict diseases, I would suggest the following two review papers. In summary, these papers show that although advanced models, such as the random forest classifier, generally perform better, simple models, such as logistic regression, usually perform surprisingly well, and often equally well as the advanced models. However, for certain types of data, such as image data, advanced methods, such as support vector machines and certain types of neural networks, perform way better than simpler methods, as we discussed in the video about image classification. We will now have a look at two multivariate statistical methods. Suppose that we like to use a statistical test to check if there is a significant difference in sepal width between the two species Setosa and versicolor, since we only have measurements on one variable of two groups in this case. We can use a simple t-test. If you like to check if there is a difference between all three groups, we can use MANOVA, which will tell if at least one group significantly differs from the other groups. Suppose that we now like to know if there is a significant difference in both the sepal width and the sepal length between, for example, Setosa and versicolor. When we have two or more dependent variables, but only two groups to compare, we can use the corresponding multivariate t-test called the Hotelling's t-square. When we have three or more groups to compare, we can use the multivariate version of ANOVA, which is called MANOVA. However, note that ANOVA and MANOVA also work on two groups. Finally, we will have a look at some regression techniques that are commonly used to analyze multivariate datasets. Suppose that we like to create a regression model that can predict the sepal length, given that we know the petal length, petal width, and sepal width. The problem is that we will have collinearity in such a model because we have a too strong correlation between two of the explanatory variables in the model. One way to solve this problem is to combine the three variables with principal component analysis into fewer variables. For example, we can combine the three variables into two new variables called PC1 and PC2, and then perform regression by using these two principal components as our explanatory variables. Such a method is called principal component regression. Partial least squares regression can be seen as an extension of principal component regression, where the combination of the explanatory variables is done in a way so that we instead maximize the prediction of the response variable. Both partial least squares regression and principal component regression are used to overcome the problem with collinearity in linear regression, and in cases where we have more explanatory variables than observations. Instead of combining the variables, we can simply reduce the number of explanatory variables where we only keep the variables that are important to explain the output. One such method is called LASSO regression, which is a regularization technique. If we would use LASSO regression to reduce the model from 3 to 2 explanatory variables, we would end up with the following reduced model. The variable pedal width was therefore removed from the model because it is not important to predict the sepal length. We have previously seen that there is a strong correlation between the pedal length and the pedal width, which means that we can predict the pedal width quite well if we know the pedal length. Only one of these two variables is therefore necessary to predict the sepal length. Note that LASSO regression can also be applied to other types of regression techniques, such as logistic regression to predict, for example, a categorical outcome. Similar to partial least squares and principal component regression, LASSO regression and other types of regularization methods such as ridge regression can also be used in a special case when we try to fit a model with more explanatory variables than the number of observations we have in the dataset. This was the end of a very long video about how to select an appropriate method to analyze multivariate data. Thanks for watching.